Well, thank you all for joining us. We really appreciate the time that you've set aside for this webinar. As the parents of disabled children, I understand really well how hard it is to find time in your day to do anything other than care for your child, so thank you for doing this. For those of you who don't know me, I'm first and foremost Andrew's dad. I'm also a co-founder and president of the KCNT1 Epilepsy Foundation. What we're doing today is part of our Pathways to Trial program, which is something that we're going to be hosting over the next 1 to 2 years, a series of events to help our KCNT1 community learn about and prepare for trials. So some quick background about the foundation for those parents and families who are new. We spent the first 5 years at this foundation as a group of volunteers, and what we focused on to start off was gathering data about our disease and making connections with scientists around the world that were working on KCNT1. Our main goal was really to attract the interest of pharmaceutical companies so that our kids and families could have a chance at a better life. We were really excited that over the years, several companies have expressed an overwhelming interest in KCNT1 and in bringing treatments to our children. Today, we have the first opportunity to meet with one of those pharma teams. We have the Actio Biosciences team here today, and we're going to learn about a drug that they are hoping to bring to trial soon. So with me today at the table, we have David Goldstein, the founder of Actio Biosciences, and Sunil Sadeo. We could not be more thrilled that you took the time out of your day to drive up from San Diego to meet with us so that you could share insights about your program. So thank you so much for being here, and I think we'll just get started. So, I think it's helpful for our community to understand, I think, the people that are interested in us. And so, David, maybe you could start with your story. How did you get into your career in genetics, and was there a personal connection, or how did you end up going down the pathway you did?
Well, first of all, it's really wonderful to be here, and I really appreciate the opportunity to talk about our program, which we are really excited to bring forward as quickly as we can. So my career started out in genetics. How I got into genetics is now just a little bit of a mystery to me, frankly. I was interested in a whole bunch of things when I was in college, including political science, history, literature, and I did some biology, and I found genetics to be a just truly exciting puzzle. Just got really, really fascinated by it. And so I was really drawn in first to the intellectual world of genetics. And because of that interest and passion for genetics as a science, I decided to make a career out of it. And part the way through my academic career in genetics, I became increasingly interested in trying to do something useful therapeutically with genetics. So I progressively paid more and more attention to medical genetics, spent a lot of time thinking about how to use the rapidly evolving science of genomics, being able to interrogate entire genomes to figure out the causes of disease in individual patients. Was involved in methodological developments, and eventually was involved in the search for the causes of disease in individual patients and the communication of causes of disease to lots and lots of patient families. And what I found when I communicated those diagnoses usually was that the story I had to share with the families was, we didn't know what the disease was before. We've done genetics. We now know what the disease is, or we know the cause of an already known disease. It's pretty bad. The prognosis is not so good, and unfortunately, there aren't any treatments. And I was doing that again and again and again. And if you think about it, it almost has to be that way, because if you look at the world of Mendelian diseases, the majority of them are not well treated. In fact, the vast majority of them are not well treated. So I made a decision after some years of doing that, that I would not spend the rest of my career communicating to families that, well, we found out what the individual had, but there isn't a good treatment. So I decided to dedicate the remainder of my career to picking a few diseases where I could help to change that. And so we started Actio to do exactly that.
It's interesting that you say that. I mean, that was essentially our experience. You know, Andrew was having seizures. We weren't sure why. Over many months, the decision was finally made to get a gene panel test. And then when we got that result, it was as if somebody had just read about it on Google minutes before they presented us the diagnosis. They presented us the diagnosis, and then the final, most important statement, which was, oh, and there's nothing we can do about it. Which, as a physician who's used to hearing about a diagnosis and problem solving and treating it, was insanely frustrating. Can you share, Sunil, with us, what was the evolution of your career that led you to this moment with Actio?
Yeah, happily. So again, also I'm very happy and honored to be here. So I've been fortunate. I've actually worked on rare diseases for the majority of my career, including during my PhD on a rare ataxia. And I think once you start working on areas of a complete unmet medical need where really every experiment you do is contributing to a field where there's no treatments, I don't know how as a scientist I could work anywhere else. So I was fortunate to collaborate with David actually when he was as an academic at Columbia, working on a collaboration on rare diseases. And he called me one day and told me what Actio was about. And David described how he helped put into clinical practice diagnostic sequencing to identify mutations that cause disease, and identified multiple rare disease-causing gene mutations, and grew frustrated going to parents and saying, I know exactly what's causing this disease and there's nothing for it, and I'm gonna go build a team and a company to go change that. And I leapt at the opportunity. I joined the same day he did, and working really, really hard with an amazing team to find treatments for devastating diseases.
So that brings us to Actio. And David, one of the things that I think you ended with was that you talked about a handful of diseases that you thought you could make a difference with. How did you figure out what those diseases are? I mean, one thing that never ceases to amaze me is, we've got 10,000 rare disease, 500 roughly genetic epilepsies, and of the rare diseases, 1%, I believe, have therapeutics developed for them. And it never ceases to amaze me that as one of those rares, how teams find KCNT1 and they have this sense that they can do something about it. How did you arrive at that conclusion?
So I was not only frustrated with the overall picture that most diseases are, rare diseases are not well treated, but what was even more frustrating is at least some of them looked to me to be tractable drug development propositions.
When you say tractable, you mean improvable?
Predictable. So
Okay.
Drug development is always very, very hard. And unpredictable in general. And everyone will tell you it takes a very long time, it's very, very expensive, and the outcome is very, very uncertain. That is generally true. But while nothing is easy in developing a new effective therapy for a severe disease, some diseases do look easier and more predictable than other diseases. And I found it extremely frustrating that some of these rare diseases looked to me like opportunities to launch a drug development program that would be very likely to succeed. And it seemed to me entirely inappropriate for it not to be happening if it was likely to succeed. And I actually spent some time trying to convince existing pharmaceutical companies to go after these opportunities, because I said, look, it's going to work. And I had reasons, and so I'll go into to say it really ought to work. And they mostly took the view that that might be true, but they're too rare to really warrant the sustained effort required. And I took the view that the fact that you're pretty likely to succeed means that not only is it a good thing to do for the families that desperately need the therapies, but it also means that it's a reasonable economic proposition. Because one of the reasons everyone says it's so expensive to develop medicines is because of the very high failure rate. But if you can say, look, here are the reasons why we probably will not fail, it makes it a much more compelling opportunity, not only to deliver a therapy, but also economically, because you're probably not going to fail, so you're probably going to get something out the other side. So I really felt that there was a business case to be made that you could really do this, as well as the desperate need for the therapies. In the case of KCNT1, the reason that I took that view is that it's one of the better understood Mendelian diseases, in the sense that we actually know how the mutations cause disease. We know they all cause disease in very similar ways, all the different mutations in the gene. And we know that the mutations are in a protein that can be inhibited with small molecules, which means this is a very well-established drug development problem, very well-established in virology, very well-established in cancer. There is no reason why you couldn't do it. And that was really one of the great frustrations, looking at this absolutely devastating disease and feeling like, you know what, this can be done and it should be worked on. So I tried to convince other people to do it. I didn't succeed, so decided to do it, myself.
I'm curious, you speak really eloquently to the science component of what makes a disease interesting. I mean, it has to be, and tractable, as you say. I'm wondering, on the personal side of it, I imagine that one thing that I've encountered over the years of interacting with scientists and pharma teams is the disconnect sometimes between what you read about our disease in a paper and what it's really like to raise kids like ours. I mean, quite frankly, even our clinicians who take care of our kids on a regular basis and are the experts in our field sometimes don't really truly understand it is what we're living with. And I know that you had an opportunity that we were we invited you and we were really flattered to have you visit us at Epilepsy Awareness Day at the Disneyland Hotel, and then we had a few of our family show up, and I'm wondering what kind of an impact did that have on you in terms of the energy that you bring towards making this disease go away, and were there any other insights that you came away with in terms of what your goals should be, the speed at which you should work at? How did it affect you to meet Andrew and our other children?
Well, really the way that it affected, I think, all of us, those that have directly interacted with you and Andrew, and those that have heard a lot about Andrew, we went away feeling like not only are we absolutely committed to the program, but it has to go as fast as is humanly possible. And it ended up being really, really important to have that experience, because there is a tension in a company between wanting to get there as quickly as you can and wanting to do the work as well as it can possibly be done. And it's a healthy tension. I mean, obviously, we don't want to race forward with a compound that doesn't do what it needs to do therapeutically or that introduces unacceptable risks. So we want a compound that is good as it can be. However, we don't want to spend years tinkering with improvements that don't make that much of a difference to patients, recognizing, frankly, how desperate the need is and the risk that there's a lot lost every year that will be very, very difficult to get back, meaning that every single month matters. And, I came back from meeting Andrew with the message to the company that we needed something that would work absolutely as fast as we could get it, and that everyone had to understand that every single week mattered. And that message now is through the entire company, and everybody is completely behind the idea of, we're going to hopefully do this in something approaching record time. And that is because we all throughout the company truly, truly care about how fast we can do this because of how urgent the need is and because the days matter to the families.
They do. I mean, each individual day is a struggle. You go to bed exhausted thinking there's no way I can get through another day like this, whether it's working your job or taking care of your healthy children. And then the insanely difficult work of keeping a sick kid like ours alive. I remember in terms of the timing, you talk about weeks and months, and that's how I've always thought about this disease. Every month we lose a child in our community, and I measure everything we do around that benchmark. When somebody tells us, well, it might take 6 to 9 months, I ask them, well, do it in 6, and if you can really figure it out, do it in 5, because those are multiple lives you're going to save doing that. But I juxtapose that now also, though, is against the experience that we've had. So, you're aware that we had a trial in our community, an N-of-1 trial for an ASO, and we lost one of those children, and one of the other children became really sick. And so on the one hand, when Andrew was initially born, I thought I will do anything in any amount of time to get my son a treatment. And I didn't, I thought I'll take on any risk. And then something like that happens where these pioneer families take on the incredible responsibility of putting their children in a trial. And so I would say that I have in some ways changed the way I process it. And what I'm wondering is, everything you say resonates with me in terms of speed, and it is absolutely deeply appreciated. What I'm wondering for families like mine and for others out there where they're feeling that same sense of angst about the balance between moving fast but also not hurting our children, knowing what you know about that program and what you're doing, could you describe what you're doing different that might give our families a sense for why this is just different from a safety point of view, potentially?
Yeah, let me back up and just say first that the appropriate amount of risk to take is a very difficult judgment. And, that's a judgment that every family has to make, and it is a hard judgment. And I have no criticism of any decisions about how much risk is appropriate to take. Some modalities are inherently riskier than other modalities, where what I mean by modality is, what kind of treatment you're using. So if you're using a treatment like a gene therapy, there's not a huge amount of experience with gene therapies. There are a lot of risks associated with gene therapies of various kinds. If you're talking about, actually introducing an entire new gene, that's very challenging. If you're talking about targeting the message that actually creates the protein that causes the disease, as happened in the so-called ASO trials, those can generate reactions to the therapy themselves, and that's a well-understood risk of those therapies. So the modality itself that you choose varies in terms of risk. Small molecules are a very, very well-established, very well-understood modality for treatments. And there is a very elaborate process you go through before you get into testing them in people to try to get a sense of how safe they are. And that process actually is pretty well established, pretty well understood, and works reasonably well. Now, can't predict everything that's going to happen when you get to people, but it does work reasonably well. And because of that, if the disease is one that can be treated with a small molecule, which is a very well-established modality, as I indicated, that's actually probably the way to do it, because you really can probably then treat more safely, as long as it can be done. Now, a lot of diseases, there isn't a clear, straightforward path to treat them with a small molecule. One obvious reason for that is for some disease-causing proteins, you can't do anything to those proteins with a small molecule. You just can't get a small molecule to bind to it, so you can't modulate it. Or maybe you can modulate it, but you can't modulate it in the right direction. So in some cases, you've got too little of the protein, and maybe it's a protein that you could inhibit, reduce the activity, but you can't resurrect activity that's not there. It's a much harder proposition. For KCNT1, the basic problem is overactivity. That's always easier to do with a small molecule. And this is a protein that a small molecule can bind to. So this is something you can do with a small molecule. On top of that, when we talk about safety of small molecules, we think about whether it's safe to act on the target that you're intending and what else you do that, the compound interacts with other proteins that are not the intended target, so-called off-target effects. In the case of KCNT1, we have very, very good reason to believe that inhibiting the target itself will be safe and well tolerated. Lots of good reasons to believe that, both from animal model studies, but also population genetic data in humans that indicate that. So we have reason to believe that it will be well tolerated and safe to inhibit the target. And if you go through all the steps that you need to go through to see whether the compound is doing anything else that's risky, and that all comes up indicating that it's safe, then you're in a pretty good position. And the other thing I would note is that unlike some therapies, some modalities, with small molecules, you will actually typically test them first in healthy volunteers and assess the safety in people that are healthier and maybe better able to tolerate something unexpected that happens. And so you have that experience before you then get into the patient community that might be a little bit more fragile. That's the path that we would follow. And if that all looks like it suggests a safe, well-tolerated therapy, I think we could actually go into the patient community with a lot of confidence. In fact, more than other modalities, I think is fair to say, that it will be safe when we get to the patients.
Well, I think that gives us a lot of information, and it sounds like we're sort of at a point where I think maybe showing us the slides that you've prepared and the data that you've been generating to help make that, paint that picture and flesh that out, I think would be really helpful.
That sounds great. We'd love to do that.
Great.
All right, well, let me start again by thanking Justin and Sarah for the opportunity to describe our program in KCNT1, and to thank everyone for taking the time on a weekend to listen to the story. I'll be walking through where we're at in our KCNT1 drug development effort. I'll start with a quick introduction to the company, go into a detailed description of our candidate that we're developing as a treatment, which we refer to as ABS 1230, and with a description of how we intend to eventually evaluate this treatment in patients and see how much it helps. We are a small, newish biotech based in San Diego. We were founded just about 3.5 years ago now. We just became a clinical stage company and are testing our first treatment beginning in healthy volunteers. In fact, now in just about a week. And we currently have a team of 40 people, including individuals with decades of experience in all the key aspects of drug discovery and development. This is just a little bit about exactly how we work. Once we have picked a program to work on, such as KCNT1, we do work in the lab to assess whether our treatments will work for each and every patient that has disease because of a mutation in the gene that causes the disease. So what that means for KCNT1 is we characterize all the disease-causing mutations and ask whether our treatments will work for all of them. We'll describe that. And then after that work, we go on to assess how well the treatments work in so-called preclinical models. So these are animal models of the disease that are appropriate in the sense that the models are due to the same cause of disease. So the mouse model has a human disease-causing mutation in it, in the mouse, and has a seizure presentation. And that allows us to assess whether we can improve the symptoms in the model after they are already there. And that can tell us whether the condition is really too developmental to be able to improve the symptoms after they're present in people. Just a little very quick overview of epilepsy caused by KCNT1, which of course is not necessary to this community. But what I will emphasize is that this is one of the Mendelian diseases where the biology is very clear. We know that mutations in the gene activate the channel that the gene encodes and cause a particularly severe epilepsy. And we know that it's rare, but that there are probably somewhere between 2,000 and 3,000 patients in the US. If you look at the right-hand side, you see information about seizure burden that was derived from the citizen effort. I certainly don't need to tell this community about how serious a challenge seizures present for patients. But what I do want to emphasize is that these and other data really do make clear that no available treatments really do much to help control seizures, really emphasizing the truly desperate need to develop an effective treatment that can control these seizures and the very, very high seizure burden that most patients face, and all the developmental difficulties that result from that very high seizure burden. So it really just emphasizes that, unfortunately, KCNT1 positive epilepsy is really one of the worst epileptic encephalopathies in desperate need of new effective therapies. We are developing ABS 1230 as a treatment for KCNT1 positive epilepsy. The intent is that this small molecule treatment could be used as a once daily oral treatment. So you take a pill or a liquid formulation once a day, with the primary aim to have the therapy reduce the seizure burden and thereby hopefully also improve developmental outcomes. Just a little bit about exactly how the mutations cause disease. As I indicated before, we know for sure that the disease-causing mutations do indeed activate the channel. We moreover know for sure that mutations that reduce activity, even reduce it entirely, do not cause epilepsy, because there are what we refer to as loss of function mutations in the human population that do not cause epilepsy. So what we need for a precision targeted therapy for KCN2 positive epilepsy is an inhibitor that can reduce the activity of this channel when it's in an overactive state because of the activating mutations, which basically means just that there's too many ions moving across neurons, and that through the pore that the channel encodes. And that actually is what ultimately leads to the excitability in circuits that cause the seizures. So we need an inhibitor that can push down the activity of the channel, and critically can do so regardless of what mutation actually causes disease. There's just a little bit pictorially about what we are trying to do. What we want to do is target exactly the cause of disease and not act somewhere else in the biology of neurons to try to reduce seizures, which is what most anti-epileptic drugs do. So what happens is KCN2 is too active, and as shown in the figure, what that does is make inhibitory neurons unactive, so their activity is reduced, so they can't dampen circuit excitability as they should, and then the circuits become excitable, and that's what leads to seizures. So the targeted therapy is just to go after the root cause, reduce that overactivity of that channel, normalize then the activity of the inhibitory neurons to conserve their appropriate function, and then that keeps the circuits from being excitable and controls the seizures. So what we want to do, thinking about all the different mutations, is look at the activity of the normal form of the channel, which we can do in the lab, and then look at the activity of all the disease-causing mutations. The cartoon of this is shown on the right-hand side, where that gray circle is representing an activity measure we can look at in the lab for the normal form of the channel. All the blue ones are mutant forms that have been reported in patients. In fact, all of them characterized at the company. And what we want to do is push down the activity of all of those mutant forms to get them back down to normal activity levels. And that's the precision therapy. That removes the root underlying cause of disease. That is what we're aiming to do. So this is where we're at. If you look at this figure, what it's actually showing is the activity of different compounds against mutant forms of the channel. If you look at the left-hand side of the figure first, you actually see the best compound that was known before we started work. And you see the activity of that compound for a bunch of different mutations that are recurrently seen in patients. And to make clear why this matters, we've highlighted Andrew's mutation. And you can see here that the activity is actually okay for some of the mutant forms of the channel. The bottom part of the figure is where the activity is higher. And for a lot of the mutant forms of the channel, the activity is very weak. That's the top end of the figure. Our compound, however, ABS-1230, has been assessed for almost every single disease-causing mutation. And the activity is very, very strong against all of them. I will just make one quick research note that just about every single mutation has been assessed. A couple of the mutations are, for technical reasons, very hard to assay the way that we assay it. But we think they act in the same way as all the ones that can be assayed. So the next thing we do is ask, well, what happens in the mouse model of the disease? So what we do is take a disease-causing mutation in humans. With collaborators, we put that mutation into a mouse model, look to see if the mouse recapitulates what you see in humans, and then actually, of course, test to see how well the treatment works. This work's done in collaboration with Matt Weston, and he has had a very long-standing interest in KCNT1, spoken with the community before, so most people will know him and his commitment to KCNT1. He's one of the world's experts on KCNT1. So we've been working with him with a mouse model that he developed for one of the mutations that's commonly seen in patients. And the mice with that mutation do have a high seizure burden, so we have good recapitulation of what happens in people. And you can actually see that seizure burden depicted for individual mice on the left-hand side, where each circle is a mouse and the seizure burden that mouse has. And then on the middle part of the figure, you can see what happens to the seizures following treatment with ABS-1230. You can see that the seizures are almost entirely eliminated upon treatment with ABS-1230. So what's happening here is the channel's overactive in the mouse because it has the disease-causing mutation from humans, so it's an overactive channel. The mouse is treated with ABS-1230, and the channel is no longer overactive. And then when the channel is no longer overactive, the seizures go away. Now, this is critically important because it tells us that the seizures are not completely what we call developmental. They're not a consequence in this model anyway of something that went wrong because of the mutation early in development that can no longer be addressed. If after the seizures are there, you do address the underlying cause, then the seizures are controlled. This is exactly what we wanted to see. When we then allow the drug to wash out, then the seizures return, making clear that it's a response to the treatment. And one of the more interesting comments that I've heard is what Matt said to us after he saw these results from his lab. He said, you know what, these are results that really change the way I think about the brain and about how KCNT1 works. So it really is what we were hoping to see, and very, very encouraging about the possibility of controlling seizures in KCNT1 patients. So there's just a little bit about the phases that you go through when you're developing a new medicine. You first have to discover a good compound. We have an exceptional chemistry team that really has generated some spectacularly good inhibitors of the target. You then do a variety of preclinical research, which you've just heard about, to really characterize the compounds that you've developed, show how they work against, in this case, different disease-causing mutations, how they work in appropriate preclinical models to try to predict what will happen when you get into patients. And then you move into the clinical stage, where for small molecules, usually, you will first look at how safe the compound is in healthy people. And then after that experience of safety in healthy people, you'll then move into a small patient study. And that small patient study gives you an idea of how well the therapy works. In this case, an idea of the extent to which we can control seizures, how much we reduce them from the baseline. And then that information will be used to determine how many patients need to be in the pivotal study that will be taken to regulators to try to get approval for the new medicine. And that's where we're at. We are hoping that progress will run as quickly as possible from here. I do want to emphasize that all phases of drug development are unpredictable. So we can make no promises about the timeline, but we are hopeful that we will be at a point of testing our therapy and assessing how well it works in patients sometime in 2026, but emphasizing again that it's unpredictable and there are no guarantees about timelines. But we are hopeful that the program will continue to move as it has to date quickly. And I can tell you that the company, every single one of us, is absolutely committed to moving this forward as rapidly as we possibly can, while still doing the work as well as it can be done. So with that, I thank you all for the time on the weekend.
Well, David, thank you very much for walking us through those slides. It's really informative. I think it's really helpful. So now we're at the part where I think we want to open the floor to questions. We've had some patients in our community, some family members submitting questions. I think that these are really important for our family members to hear the answers to these. We've been talking to our communities for now months about how they feel about trials. So we want to make sure that their fears, their questions, their concerns are all addressed before we, get to the point where there may be a live trial. So with that, we'll start with this first question. What is the primary goal of a clinical trial that you're working on?
So what we're trying to do in the trial is assess how well the treatment works. We want to find out whether the treatment really can, as we hope and in fact expect, whether it can really control seizures.
So the next question we have, it seems very simple, but I feel like to me there's a lot of layers to it. How could a trial like this benefit my child directly? And I say that on behalf of all the parents who are listening now and who may listen to this, as we record this and put this on our website for the future. That's what we all want to know ultimately, is what can we expect out of a trial like this for our kids?
Once we start treatment in a trial, if it works, our intention is to have all of those individuals continue being treated. So the benefit to participation, the direct benefit to participation, is the possibility of getting an effective treatment earlier. We really are hopeful that it will work. We recognize how important it is to have patients treated as soon as possible. And participation in the trial will mean treatment much earlier than otherwise, because for those that don't participate in the trial, treatment will have to wait until there's an eventual approval, which will take some time.
So when I think of Andrew and the thousands of seizures that he has, given what you've shared with us today, the slides that you've talked about, the science that leads up to this potential use of this medication in an actual human, you really feel strongly that patients like Andrew will see a reduction in their seizure burden and see a better version of their life?
We're doing this because we genuinely believe that this is the way to substantially reduce the seizure burden. It's hard to predict what's going to happen when you get into people, that's true. But everything that we know now suggests that the treatment really will substantially reduce the seizure burden. And moreover, things that I've heard from you and others, I think give us reason to believe that in reducing the seizures, developmental outcomes will be improved too. Even just the experience that those periods of time where seizures are better controlled, you can see the child come back to a degree, strongly suggests that the ongoing seizures just make it extremely difficult for the children to develop. And if we can get in and substantially improve the seizure control, it should also lead to improvements in the development of the children too.
Okay, so for the changes that we've seen in Andrew have come from a combination of this laundry list of drugs that our clinicians try on us, ketogenic diet, and other interventions. So if those are drugs that were not really made for KCNT1, it was sort of, here's some epilepsy drugs, your child has a version of epilepsy, we're just going to throw these drugs at your child one at a time, then two at a time, then five at a time. We have seen some small changes in Andrew's development with those sort of reused drugs or non-direct therapeutics. Is it fair to say maybe that a drug that was purpose-built for this disease could have potentially a bigger impact on the developmental changes that we've seen so far?
I think that there are two primary reasons to believe that a purpose-built drug has a better chance of improving development. One is, as a precision therapy, there's a very good chance it will simply work better to control the seizures than anything else. And all the data that we have so far, including the results in the mouse models, including the experience of ASOs in patients, all those data really do suggest that going to the underlying cause of disease, which is the overactive channel, and normalizing that overactivity is the best approach to control seizures. So we have good reason to believe that it will do better than anything else at controlling seizures and have the benefit of development of controlling seizures better. But beyond that, mutations that cause disease often directly do more than one thing. In the case of KCNT1, it might be the case, we don't really know for sure, but it might be the case that some of the developmental problems are a consequence of the seizures, and some of the developmental problems are a consequence of the disease-causing mutation, but not working through seizures. Those developmental consequences, they would also be improved by the targeted treatment in a way that a treatment that is just simply suppressing seizures cannot do. So those two reasons give us optimism that this is really our best chance at improving not only seizure control, but developmental outcomes too.
Okay, thanks for explaining that. I hadn't really thought about that. Most of the folks that we talk to tend to talk about, well, the seizures make it hard to develop, and in theory, if you can stop the seizures directly, that you'll see improved development. But the idea that it could be, the mutation could be working in two ways, both to cause seizure and in its own separate path, parallel to that, also causing developmental issues, means that if your drug works, and if it's safe, you could potentially target both of those, not just one leading to the downstream effect, but actually targeting them both directly.
Exactly.
But and it's something I guess we don't know until we know.
We don't know until we see.
Okay. This next question, how soon would you expect to see changes in someone taking your drug?
So based on our preclinical model results, we expect it to be very fast.
Can you quantify that? I mean, I don't want to hold you to an exact number, but as an example, we saw improvement in Andrew on the ketogenic diet 2 or 3 months down the road. And so we had to initially it felt like a failure, like every other drug we'd ever started, because there was never an immediate change in any kind of capacity until keto, but it was quite delayed. And I think our doctor at any given point could have said, well, we tried this for 6 weeks, let's stop this. We could have easily said that, and thank God we didn't, because eventually we saw this dramatic change.
Yeah.
So are we talking days, weeks, months, or is it one of those things that we just don't know, again, until we know, because we've never used this drug in humans?
Well, so as David described in the presentation, in the preclinical model that we've used with a human disease-causing mutation in KCNT1, you see reduction in seizures within days.
Okay.
Did you want to add to that?
Yeah, the only thing I'd add is that to the extent that we understand what's actually happening with the brain circuits, there is an expectation that it would be rapid. And the reason I say that is that, as I mentioned in the presentation, we know that the overactivity of this channel directly reduces the activity of inhibitory neurons. So we know that that's what's happening in part to cause disease, maybe in whole to cause disease. We don't know that, but at least we know it's contributing. And we know that when we introduce an inhibitor of the channel, that actually should increase the activity of those inhibitory neurons very quickly. Meaning actually in terms of changing the activity of those neurons in hours. So we should have an impact on those circuits relatively quickly, and that should actually translate to seeing seizure control in a matter of days. The caveat there is the very obvious one that mice are not people, and it's hard to know exactly how predictive these models are of what's happening in people. And there is an expectation that there are some developmental aspects of the disease too, that the disease-causing mutation has some direct effects, and then it may change development in some ways. And some of those things might lead to slower responses to treatment. And the balance between the more immediate responses and the things that are slower because they're developmental aspects, or other things that take time, that balance is hard to predict in advance. But based on our understanding from the preclinical models, from the underlying physiology of what we think is happening in neurons, there is reason to be hopeful of a rapid response, and that's exactly what we'll be looking for in the first early trials.
Okay. So if I sign up for a trial and I get into a trial somewhere and we put Andrew on this drug, 48 hours later, we don't see a change. Let's say worst case scenario, we don't see a change in seizure burden like you've seen in the mice where that curve just flattens out so dramatically. Because it's humans and mice are not humans, I have to be patient. You know, I'm hearing this presentation, you're sharing the data like everybody else in the community. We have to be, I guess, remind ourselves that that's not necessarily what we can expect, number one. So I guess this is probably not a fair question, but I guess are you building into the trial sort of an endpoint where you say, hey, if a family doesn't see or a patient doesn't respond either to the seizure component, let's say, by X amount of time, you might stop it, or is the idea to go through the entire trial? And I guess the other part of that is, like you addressed, the development is maybe even more likely to have more of a delayed response. And that could be, even if you don't see the seizure change, that is still a reason to say, hey, you want to stay on this for the duration of a trial because A, there could be a lag in both of them. B, it's also possible, I suppose, that you could have no change in seizure burden but still have a demonstrable change in the behavior. Is that possible?
Yes, yes, for sure. So these are absolutely important points to make. So first, we want to be prepared for the possibility that seizure control in patients turns out to be as good as the preclinical models suggest. And if that's the case, we want the most rapid path possible to getting the medicine approved and available to everybody. So we want to prepare for that possibility. However, it would be entirely wrong to give up on the treatment because we didn't see an immediate response, because Patients are not mice because we may be asking too much of the model to predict not only whether it's going to work, but the time frame over which it works, and because it is a precision therapy that undoes the underlying cause of disease. And if you think about what's really happening, the bottom line is actually really, really clear. The bottom line for this kind of precision therapy is it has to work for any aspect of the disease that isn't entirely developmental. So the way to think about it is the mutation is an insult to the development of the individual. It's something that goes wrong, and it can have ongoing consequences, but it can also have consequences that are entirely developmental. So something responds to that insult, and then it itself causes the problem, the response, the developmental response to the insult. And then if you go and undo the underlying cause, the developmental aspect, you're not treating. It is possible that part of the presentation, and it would actually be the expectation, that part of the presentation has that kind of developmental aspect to it, and part of it is an ongoing effect of the disease-causing mutation. So if we go in and after 2 or 3 days we don't see improvement in one aspect of the disease or another, it doesn't mean that other aspects won't be improved over time, ones that are harder to see immediately, like ability to learn. So as a precision therapy, we need to give it time to see how it helps. We are extremely hopeful that it will help with seizures right away. But we do have to all recognize that there's only so much that those mouse models can tell us. And so we need to go into the trial prepared for things to turn out as well as we hope, but also prepared to keep looking longer to see how the therapy helps if it doesn't control seizures as fast as we hope.
Okay, that makes a lot of sense. Thank you. More simple question, I guess, probably, but how often will kids need to take this medication?
So based on the data we've generated studying this drug, we believe once a day will be possible in patients.
Okay, that's pretty easy. I mean, a lot of our kids are taking drugs 3-4 times a day, so that would be a nice change of pace. I think this is something that every parent listening can relate to. I mean, we know from our data, from the participation of our patients in the Citizen program, that 95% of our kids who are 5 and over are on 5 different drugs at the same time, which is, it seems wrong. You know, it's a challenge just to distribute meds like that into your child. I mean, when we get Andrew ready in the morning, we line up one bottle after another, and we confirm the dose before we give it, you know, because we're chronically exhausted, and last thing you want to do is either miss the medication or give the same medication twice. It's a laundry list of drugs. It's exhausting, and there's so many opportunities to get it wrong when you're chronically deprived of sleep and you're distracted by your other children. I think the ability to take, you know, a pill a day, is beautiful, but in terms of, you know, stopping this laundry list of medications, is there anything we should do to be prepared for this, or can we just basically show up on the regimens that we're on and enroll in a trial?
You can show up without changing the medications that the patients are on and enroll in the trial. And then what we would be looking at is to see whether adding in the new treatment improves seizure control. The only thing that you'd need to do is not change the dosage of the other medications during the trial, so that it wouldn't have the complexity of whether the change in dose had an influence on the control of seizures. But it's important to emphasize that if we show that the treatment works and it eventually is approved and it substantially improves seizure control as we hope it will, then what care providers would do is slowly remove the other medications because you're getting seizure control out of the new treatment. We know that anti-epileptic drugs themselves actually cause a lot of problems with development, and so there would definitely be some urgency to begin removing the medicines that are no longer necessary to control seizures.
You know, another thought that comes up as I see a question like this about stopping medications is, one of the common denominators with a lot of the drugs our kids use is that they're highly sedating, and we have sedative on top of sedative, whether it's benzos, barbiturates, and no adult would want to take those safely. It's the kind of thing you read about in a newspaper when somebody overdoses. It's these combinations of drugs that are really pretty dangerous, and our kids are on these every day. Sedation and the having children who are often sleeping throughout the day and not interacting with us is, I think, emotionally a big issue in our community, because we don't see children opening eyes and looking at us. And I'm slowly getting my point, which is the question: do you have any, given the type of medication that you've built, the pharma properties of that medication, is this a medication that we are going to also see sedation with, or is it in such a different category that we think that we might get kids who are not only having better seizure control but are more alert?
We have absolutely no reason to think that this treatment will be sedating. We have no reason to think that. And this goes back to the point I was emphasizing about the difference between a targeted treatment that's targeted to the exact cause of disease and a treatment that is simply trying to suppress seizures. Most of the, not all, but most of the anti-epileptic drugs, they are sedating, and what they're doing is they're just quieting neuronal activity, which is why they're sedating and why they reduce seizures. So they're suppressing seizures by reducing overall neuronal activity. This treatment is not doing that. This treatment is targeting exactly the cause and is essentially normalizing that underlying cause, moving what's aberrant back to normal. And as far as we can tell, there are in fact not adverse consequences of that normalization. In fact, moreover, even if you will overshoot a little bit, if you take KCNT1 and use a dose of the treatment that pushes it a little bit below the normal levels of activity, we have reason to believe that that won't be harmful, sedating or otherwise. And the reason we say that is what I refer to as the population genetic data. There are people in the general population that have a mutation that knocks out the activity of the channel, and those people are completely fine. There's no natural selection on those kinds of mutations, indicating that not only do they look fine, but natural selection, which I like to say has a very fine eye on what's a problem, doesn't even register that. So it's a good indication that the activity on KCNT1 of the treatment should not cause the children any direct problems. You never know that for sure going in, but there are those indications that that shouldn't happen, and it shouldn't have the sedating properties of anti-epileptic drugs.
Yeah, and just to add, in the preclinical studies we've done, granted they're animals, we've never observed that.
Okay. So in theory, if the drug that you have gives us good seizure control, we could start taking away medications like most of us have been through, where we start 3 meds, one doesn't work, they don't like it, they change the combination. But we could eventually get good seizure control and start slowly weaning off other drugs and find ourselves a child who's potentially, in theory, not only has better seizure control, but is now more alert because they're not on these highly encephalopathy-inducing medications. So fewer seizures, a more awake child, and then in theory, that more awake child might have more capacity to actually interact and learn.
Oh, this is what would happen for sure. So if seizure control is substantially improved with the new treatment, epileptologists will take the children off those other meds quickly. And they will be extremely eager to do so. We've heard that directly from many of the people that care for these children. They know how damaging. It's just that, they're desperately trying to control the seizures.
Yeah.
But if the seizures are controlled, then yes, they will come off the other meds quickly.
Okay. I have a child on a ketogenic diet. Will this medication interfere with that?
Oh, that's a good question. So medicines have an active part, and in order to get to where it needs to get to in the body, you have to package it sometimes with other ingredients, including sometimes carbohydrates and sugars. That's called the formulation. So our goal for ABS 1230 is to develop a formulation that doesn't require those and is completely compatible with the ketogenic diet.
Okay, that works. I mean, I would say that anytime that we go to the ER or anywhere else, we bring our binder that says, please don't add sugar to anything, whether it's the saline that they're getting. But we know that we're getting exposed to some. I think as long as it's not a tremendous amount, and if you've got a drug that's super potent, I assume that it's a really small number of milligrams that they're getting. They're not getting a 500 milligram tablet, so it's only going to be a fairly limited amount of carbohydrate, even if you have to have that in there. Yep. But it's nice to think that you could formulate it theoretically without any. But I think that our kids are they're fairly, I think, resilient to having a little bit of exposure. And in fact, sometimes we get too much acidosis, and we start dropping orange juice and sugar to kind of, to break that. But that's good to know. How long are you, so is that still, pardon me, are you still in the active phase of, is formulation going on now? Or, okay. Is that a fair question?
Totally fair question. Okay. Yes, we don't know yet what the patient formulation will end up being.
Okay. Okay, so my child is non-peroral and uses a G-tube. Can the medication be crushed if it's in a pill form?
We are very aware of which is one of the hugely important roles that the foundation plays, making us aware of the patient population that we are intending to treat. So we're very aware of the feeding tubes, right down to knowing the manufacturers for the ones that are commonly used. And we are testing our potential formulations in awareness that they will have to be compatible with those tubes. Okay. So that is explicitly part of the plan.
Okay. Is there a decentralized trial option where some visits can be done remotely? So for anybody in the audience who hasn't been following trials and maybe used decentralized as a common word, the idea is, can we do it remotely? So, some trials, parents and the patient have to show up on site to a hospital, some big academic center. Maybe they have a relationship there already, maybe they don't. There's a lot of new things happening, and I think it can be really stressful. Is that going to be a big part of what we do, or are there going to be opportunities for us to do some of the trial from our homes where our kids are most comfortable because they're really sensitive to their environment?
The majority of the information we collect for the trials will be collected at home. So we will, in the decentralized terminology, we will make the trial as decentralized as we possibly can. We will be absolutely minimizing visits to a hospital or a clinic. The exact design is not yet settled, but the primary information that we are collecting will be seizure diary information from the families, some use of at-home EEGs, and a very small number of visits to a site that is selected to minimize the travel burden. So we are very, very aware of how difficult it is for families to travel, and we don't have the design finalized, but we are pursuing a design that will minimize the required travel.
How much of that is determined by your team based on your best sense of what suits our community, and how much of it is influenced by the FDA when you submit your application to say this is what we're hoping to do for this community? Can they push back on whatever it is that you design?
Yes, they can push back, but I don't think that in this case there's going to be much of a challenge, because there's already regulatory precedent for using seizure diary data as an endpoint. So we believe that we will not get pushback.
That's great. What countries do you expect this trial to be available? We have communities, like we said, over 50 countries around the world. We have lots of really interested patient advocates who are struggling all over the place. And our intention years ago when we organized this foundation was to try to be a voice for the worldwide community. So to that end, how do you have a sense of what the progression of your trials would be, whether it's starting in the United States, and are you going to start trials in multiple countries at the same time, or is it going to be starting one and then adding a second, adding a third over a 3 to 10 year period? What's that look like?
It is our intent for the treatment to be available everywhere. The exact sequence of activities in different countries is not yet determined. The expectation is that we will start first in the US, and then with a small lag, roll out some other countries. The exact countries that we go to next and the timing is not yet settled. But the expectation is begin in the US, move relatively rapidly into testing in some other countries too, and to eventually, as quickly as we can, have the therapy available everywhere.
So I don't know about other families in our community, and without getting into the politics of it, my wife works for the FDA, and not on the drug side, but she works on the device side. My wife's an ophthalmologist, and for a lot of reasons, mostly about taking care of Andrew and keeping him alive, she stopped practicing clinical medicine, and when we finally got to a point where she could breathe again, she wanted to work but didn't want to go back to seeing patients. So she's been on the device side now for the last 2 years, and as recently as 2 weeks ago, she saw half of her team get laid off. And as soon as I heard that, I thought, well, are there similar ramifications? I mean, it's two different branches. You've got the drug side and the device side. And I thought, well, what are the ramifications for the drug side when we have this kind of amazing moment for KCNT1 where after years of no good options and losing children, we could now have a drug that might potentially be incredibly beneficial, only to not have the ability to have a review or a trial begin. You may not have all the answers, but I'm wondering, can you sort of address how your team is thinking about the ongoing changes at the FDA, and are there workarounds? Like, what's really going to happen, do you think?
I'm afraid that we don't really have any answers, let alone not all the answers. We really don't have any way of knowing whether there is going to be a challenge with the expected time frame still being met at the FDA. I mean, all I can say is I really, really hope that the critical work that the FDA does in reviewing new medicines continues to function as it has. I can only hope that that's the case. I will say that if that really proved problematic for any reason, there are alternative paths that a company could pursue. I'm not saying we're pursuing those, but there are other countries where you can actually test medicines first. You don't have to start in the US, even if you're a US-based company. Many companies test their treatments first in Australia or in New Zealand. That's a very well-established path. It is our absolute commitment to take this program forward, and we will take this program forward. I'm very hopeful we'll be able to do it as expected with US regulators. If we have to consider other alternative paths for whatever reason, we will consider alternative paths.
You can address the question about patient participation and why that's important? I mean, what do you learn from patients, and are there things that our community does that helps move along the process? I mean, I would say I've shared this with you over and over, and you've met Andrew, and you've held a very heavy, 6-year-old child who wasn't able to really support himself. So you have a sense of how we live. On top of that, we ask our community all the time to participate in these studies, to answer questions for us, to be part of these registries. And, they're including myself, we're doing things I've never done as a physician. I've never participated in a registry. I had to learn what the word meant when somebody said natural history study. We're answering hours and hours worth of hundreds of questions, some of which just seem not so relevant to our child's disease, because it seems like they were made for kids who are way less sick. Is any of this important to you, the stuff that we ask our patients to do?
I think all of it is important. We would not be able to run the program that we are running right now if we didn't have the information about the disease that has come, well, primarily, frankly, through the efforts of, your foundation and a very few people like David Bearden that have been very committed to better understanding KCNT1. Without that, we wouldn't know enough about what the disease presentation is like to know that we could run a trial. We wouldn't know that there are enough patients out there to run a trial. We wouldn't know that there are enough patients to support a market, to convince people to put money into a drug development effort. We need to know all of that to launch a serious drug development effort. Even if it's something tractable, as I said, I think KCNT1 is, it is still a very expensive, daunting proposition to develop an entirely new therapy. So the more you know about the patients, the more you know about the disease presentation, the better case you can make that it makes sense to launch and all that effort to develop a therapy. So everything that you guys have been able to put together, it gives us knowledge about the disease, that gives us certainty about where the patients are. That's all critically important to being able to actually run a program. After you run a program, to try to develop a new treatment, you need to test it to see if it works. And I will say, biggest point about patient participation is the obvious one, but the all-important one. The only way we're going to have a new therapy developed by us or by someone else that completely changes the lives of these children, the only way now is by patients participating in a trial of a new treatment to see how well it works. That's the only path here. So to really change this disease for the patients and the families, we have to have participation in trials to see if new treatments work. There isn't any other path. And so that part of patient participation is what is all important. Families have to choose what treatments are worth actually testing in their children. And that's a hard decision. But selecting the right ones is the way that we're eventually going to have an effective therapy introduced for KCNT1 positive epilepsy. And that's going to depend on families deciding to participate in the right trials.
If this was your child and this was your life that had been so severely compromised, would you take this drug that you have made and would you give that drug to your child? And what would be your hope and dream for the best case scenario for that drug?
This treatment and this category of treatment, a small molecule that inhibits the different activating mutations, this is exactly what I would do if I had a child with KCNT1 positive epilepsy. There's no ambiguity in my mind about that at all. And in fact, being able to develop something like this is really one of the primary reasons, the primary reason that I left my academic career to start Actio, because I felt that this was exactly what should be done. I wasn't at a big small molecule company to say, okay, let's go ahead and use what we're good at for this disease at all. I was thinking about KCNT1, and I had the view before and now that exactly the right way to treat this disease is to develop a small molecule inhibitor that undoes the effect of the disease-causing mutations. That's the right way to do it. I've thought that all the way through. I think that now, and that's exactly what I would do if I had a child that was affected. A small molecule inhibitor, whoever had the best one like ours currently, that can safely be administered at a sufficient dose to push the activity of these activating mutations back to normal levels. That's the targeted precision therapy. That's what I would do.
So would you let your child be the first person in the world to take this drug?
Absolutely. No, without any hesitation. For a disease like this, I would be willing to try riskier things too. But we actually think this will be a pretty safe thing and do exactly what needs to be done to control seizures. So for sure, I would, knowing everything I know, and I always have thought this, I would make the decision in a heartbeat. This is the right way to treat your child.
Well, I think that gives me a lot of, I guess, gut information, right? You've presented, I think, scientifically all the reasons why a drug like yours should work and why your drug specifically should work. I think hearing the dad side of the equation, the parent side of the equation, I mean, that to me, at the end of the day, I try not to do, I always tell my patients, I won't take you to a surgery center where I wouldn't let my own child be put to sleep. I don't think you should expose somebody else's family member to a risk that you wouldn't accept. And I think that, so hearing that from you makes me feel really good about it. I want to thank you both for your time. I think it means a lot to me, getting to know you over the last couple of years, and the way you've made yourselves accessible to us. Not every company is willing to do that. I think it speaks a lot to your commitment to what the disease really is, as opposed to just, it's not just a series of letters and numbers. I mean, we're humans and these families that are massively impacted. So the time that you've put into the non-scientific side of this is I think really important. I think it helps build trust, and I feel very fortunate to have a team like yours working on behalf of our community. So thank you so much. I really appreciate your time.
Thank you.
Thank you, and the foundation. You really have inspired us to do the work better. We truly appreciate it.
That feels like a good place to end.
Yep.
Thank you, David and Sunil, for doing that with us. We have, I think we addressed some of the big picture questions, and we've had some of our families posting questions in the chat that we'd, maybe like to take an additional maybe 10 minutes to go through. And if there's any questions that we don't address, we can always, we'll make a point of following up with families via phone calls or emails. One of the first questions that stood out was, do we know how the age of a patient will influence the response that we see to the treatment?
We really don't, actually. So one of the things that we're interested in doing, and Matt's on the call and may want to make a comment, but one of the things that we're interested in doing is looking to see in the mouse model whether the degree of seizure control that we get differs depending on the age of the mouse. That's probably really the only thing we can do right now to explore that question. And that would probably be worth doing at some point. But right now, all we really know is that we do think that normalizing the overactive channel will help in seizure control at any stage, but we haven't directly investigated experimentally. Matt, I don't know if you want to add anything to that. Or if he's even able. Oh, sorry.
No, yeah, I'm here. Great presentation, and that's something that we're interested in. We haven't started experiments with this compound in young mice, but we have been looking at the mouse model and at ages, trying to figure out exactly when the seizures start during development. So if that work goes well, that is a question we could use the small molecule to address some of that. And I think it's very interesting, and it could be informative, but we'll wait and see in the patients, of course.
All right, this is not part of the Q&A, but since Matt spoke up, for the community, I don't want to embarrass you, Matt, but there's a handful of people in our community who have been incredibly helpful to our families, and Matt Weston is one of those people that most of you will never meet. He's a brilliant scientist, has been incredibly dedicated to KCNT1. And Matt, we honestly can't thank you enough for all the work and commitment that you've put into this disease. So thank you for being here, and thank you for everything you've done over the years, and your willingness to help all the teams that are trying so hard to be such a great collaborator with all our scientific teams and our pharma teams. So thank you, Matt.
Thanks, Justin. Yeah, I came out and got to meet Andrew as well a couple weeks ago. It was great, and I have a whole lab of people that are working hard and also depend on government funding to do their jobs. So I'll just say that as well. and thank you, Justin. Justin, the foundation's, I guess, played a good role for us in terms of connecting people and scientists, and has helped us a lot in that way too. So thanks.
David, is there any, how would we assess, is there any potential for our children who have been so profoundly impacted by this disease to maybe get back on track to something that looks like normal development in an ideal world? Is that possible, do you think?
It's hard to know exactly how much development will be improved following seizure control. It's just really difficult to know that. I think what we can say, as I said before, that there is reason to be optimistic. There'll be improvements. And we can say further that the earlier you treat, the better the outcome is going to be. There's going to be damage that results from the ongoing seizure burden. The longer that goes on in individuals, the more cumulative damage there is. And so I think once there's an effective treatment out from us or from anyone else, once there's an effective treatment out, there becomes a real urgency in the community to figure out a way to get diagnoses as early as possible and treatment initiated as quickly as possible. And so I really applaud, for example, the efforts to get KCNT1 into newborn screening. Applaud the efforts to really push hard the deployment of diagnostic sequencing whenever there are unexplained seizures in children. You've got to get the genetic testing done. You have to find out what the cause is and get the patients on the appropriate treatment. The bottom line answer is we really do have every reason to think the sooner the better. And I would be hopeful that patients treated relatively young would see really quite dramatic improvements over what the course would have been without effective seizure control.
Just to follow up with what David alluded to, I think one of the biggest contributions that the foundation will have made for our community this year or going into next year is given how close we are to clinical trials, we were able to submit and get endorsed to have KCNT1 added to newborn screening panels, which means ending this insane diagnostic journey that we go through. I mean, I don't recall all of your stories, but Andrew was probably 6 or 7 months old, he probably had 20,000 seizures by the time somebody thought of getting an epilepsy panel for him, which is absolutely disgusting in my mind as a clinician. I mean, he should have had one after his first seizure, like every child in this country, single unprovoked seizure, you automatically get a gene panel test. And so these families that we meet who are waiting months and sometimes years to get a diagnosis is horrifying. So to your point about treating as early as possible, very soon, assuming we get approval, we will start showing up on the newborn screening panels, which means our children will be treated within days of being born and will essentially never go down this pathway of KCNT1 related epilepsy. They'll have the genetic mutation, but they will not express a phenotype of seizures. I mean, we hope of seizures and developmental delay. We have another question, and we may not, I don't know if you can address this or not yet, but there's a question about what is the process of a trial, and in terms of, do we have a sense yet of who can sign up, what the criteria are, or do you think that's something that we'll have to do a follow-up if and when you get FDA approval for a trial?
And so we don't have the inclusion/exclusion criteria determined yet for the trial. And I'm sorry, that's something you hear from companies all the time, and I'm sorry to say the same thing. We don't have it exactly determined yet. But what I can say is that we will have in high level general terms, we'll have a very inclusive trial. So most patients that have a disease-causing mutation in KCNT1, we do expect to be able to be in the trial. We do hope to have the exact criteria mapped out relatively soon, but my expectation is that the majority of patients that do have ongoing seizures at some level and a identified disease-causing mutation in KCNT1, that they would qualify for the trial.
We spoke about during the recorded session, we were talking about where trials could start and then how they could spread around the world with your intention of treating every patient diagnosed on the planet. We've had patients, we've seen this in the past with other rare diseases where there is limited access and families move, to wherever trials are. So I think to the extent that you're able to, I think the community would certainly appreciate ongoing information as you guys have that information to share with us so that we can disseminate that to our groups of families saying, hey, we understand that they may be getting ready for a trial in France soon, and then it might be Canada. And so to the extent that you can kind of keep us appraised of that, we'd certainly love to be able to update the community so they have a sense of, if is a trial coming to their home, or should they be thinking about relocating if they feel that cost benefit makes sense for them and their child. So
we will do exactly that. We will keep you informed as those plans develop.
Another question came up. I think you might have addressed this in the PowerPoint, but the question, just to reiterate, is There was a question, can you over-inhibit KCNT1, and what would that look like in a mouse or in a patient? If you want, if you can give like a really quick response to that.
Yes, I won't do my usual meandering responses. So we really do have good reason to believe that it will be well tolerated, even if we overshoot in inhibition. So I mentioned a few things, including the presence of loss of function mutations in humans, but also just to add detail from the animal model studies. We have looked at a number of compounds that very strongly, a number of different compounds that very strongly inhibit KCNT1 and used them at very, very high exposure levels in these animal models where we're essentially turning the channel off completely. And we actually don't see adverse consequences of that. So there are really, really good indications that overshooting won't actually be a problem. Now, I keep saying you never know until you get into people, but all the indications are that overshooting will be well tolerated.
And then if you could just reiterate your point, a question came up in the mouse models, you had the mice having a high seizure burden. Every model that you've tested in saw basically that seizure burden go into what looked like next to nothing across all the variants that you tested it in. And then at some point there was a rebound. What was can you just restate, to answer this question, how long it took before mice started looking like their mutated cells off treatment?
So it depended on what compound we used, because some of the compounds take longer to be removed by the body than other compounds. So the time for the seizures to come back actually just really follows how long it takes the drug to, refer to as wash out. So when there's not enough drug around anymore, then the seizures come back. So it really is a direct response to the presence of the drug, the return of the seizures, presence or absence of the drug.
That brings us to sort of the end of our section. We're trying to be, mindful of everybody's intense caregiving, requirements that they have every single day of the week. So thank you all for taking time to do this. Sunil, David, everybody else who joined us. Matt, thank you for, participating as well. Without giving you any kind of warning that we might talk to you live here. But, thank you all for doing this. We really appreciate everybody's interest. Like I said, or like Sarah started at the beginning, this is a first part in what we hope will be an ongoing series, which is an effort to make sure families feel educated and empowered to make good decisions about participating or not in clinical trials. So, it doesn't end here. Our hope is to have an ongoing dialogue moving forward. You can always reach out via email. I think you have, all access to all of us through email or phone, or we know most of you. So please reach out with any following questions that maybe you didn't feel comfortable asking. And we'll look forward to having Actio back. We're really hopeful that you'll be submitting this to the FDA soon, and that we'll get approval, and that we'll have a follow-up webinar to talk about an approved trial, what that really means for us, and start talking about the nitty-gritty of what's it mean to actually go from this concept of a trial to getting into and participating in a trial. So thank you all for your time. We really appreciate it.
Thank you very much, Sarah and Justin.
Everybody enjoy your weekend. And thanks also to Jason Lavin at their studio and all of you parents for attending. If we didn't get to your question in the chat, we'll be sending out, we'll be addressing it via email. All right. Thanks again, everybody. Have a great weekend.
Everybody. Thank you.